So once you have this emulsion droplet created by the bile salts absorbing onto the surface of the lipid droplets, it's then hydrolyzed by the lipases that we've talked about, the enzymes that we talked about. It's hydrolyzed, the triglycerides are broken down, and the emulsion droplet gets smaller and smaller and buds off, forming a micelle. So you have an emulsion droplet. The surface triglycerides are hydrolyzed by the lipases by using the colipases to anchor on and break down the triglycerides. And so once these triglycerides are hydrolyzed into fatty acids, they're replaced by ones from the core. Because remember, the core contains the triglycerides and the surface contains the polar heads of the bile salts. And when the bile salts build up further on the surface, you get a multilamellar vesicle. Now, multilamellar vesicles, that means multi-layered. And that buds off from the emulsion droplet. And when you've got a further bile salt build up on that and a further digestion, further hydrolysis by the lipases, you then get a unilamellar vesicle, so single layer. And ultimately, you're left with a mixed micelle, which is bile salts plus lipids. So we can see that here. You've got an emulsion droplet with multi-layers, and the bile salts build up. The surface layers are hydrolyzed by the lipases, and this vesicle buds off, this multi-layered, multilamellar vesicle. And when you get further bile salt build up on it, further hydrolysis by the lipases, you then get a unilamellar vesicle, which you can see here, single layer. And finally, you're left with a mixed micelle of bile salts and lipids. And when you've got the micelle with the bile salts and the lipids, you want to absorb the lipids into your body, and the bile salts, you want to leave them behind. So what happens is that the products of lipolysis need to cross, in order to be absorbed by the body, they need to cross 3 layers. And they need to cross the apical membrane by 3 methods. So let's talk about how that happens. Well, the lipolytic product in the micelles, which is the fatty acids, first of all, they cross the mucus layer. Then they diffuse through the unstirred water layer. What the unstirred water layer is, it's a layer of fatty acids on the enterocyte surface that's unstirred, meaning it's not moving around a lot. It's just a layer of fatty acids, and the purpose of that is to create a concentration gradient of fatty acids into the enterocyte to keep them diffusing into the enterocyte. So they cross the mucus layer first of all, then they diffuse through the unstirred water layer. Then they reach the apical membrane, so the apical membrane the lumen and the apical membrane of the enterocytes. When they reach the apical membrane, they encounter a low pH. Well, why do they encounter the low pH? Well, if you remember back to absorption of water and ions, one of the ways in which sodium ion is absorbed into the body from the gut is sodium-hydrogen exchange at the brush border. So this is an antiporter, so sodium is absorbed into the body and protons are pumped out to neutralize the bicarbonate secreted by the pancreatic duct cells. So you have this exchange going on and you've got protons secreted, then there's a low pH. So when the fatty acids reach the apical membrane and they encounter the low pH, they get protonated, and by ionizing them, essentially, that allows the absorption. It results in active carriage, incorporation into the membrane, as well as non-ionic diffusion. And by these 3 methods, it crosses the apical membrane of the enterocyte and enters the enterocyte. And now one thing to pick up upon here is that when you've hydrolyzed the triglycerides and you form the micelles of bile salts and lipids and fatty acids, you've got short fatty acids as well as long fatty acids. And the short fatty acids, they're soluble, so therefore they efficiently diffuse through the unstirred water layer. Whereas the long fatty acids, the long-chain fatty acids, they require more micelles because they need to be broken down more, first of all. And secondly, they're not soluble. So they're inefficient in diffusing through the unstirred water layer, which is summarized here. So short and long chain fatty acids, they're either soluble or insoluble. Soluble is the short ones, insoluble is the long ones, so they diffuse through the water layer differently. So these remaining bile salts that are left behind when the fatty acids cross the three layers, the mucus layer, unsteady water layer, and apical membrane, well, what happens to the bile salts? Well, the remaining bile salts enter the enterohepatic circulation, so from intestines to liver, in order to keep going round and keep forming these micelles to allow the fat absorption. So the remaining bile salts, they're absorbed at the terminal ileum and colon, because remember, you need the whole of the small intestine for the fat absorption. So at the end of the small intestine, the end of the ileum, the terminal ileum, and the colon, they enter the portal system to the liver, which is the enterohepatic circulation. Now, once the fatty acids have entered the enterocyte, then you're left with either short-chain fatty acids in the intestinal cell or long-chain fatty acids. Now, the short-chain fatty acids, they move straight into the blood, whereas the long-chain fatty acids, including cholesterol, phospholipids, and monoglycerides, so now what happens to them is in the intestinal cell, they're re-esterified. So they're turned back into triglycerides. So you can see here, so they're once absorbed by enterocytes, short-chain fatty acids move into the blood, and long-chain fatty acids are re-esterified into triglycerides. And for this reason, short-chain fatty acids are suitable fat substitutes for feeding patients with malabsorption. You can feed fat malabsorption patients short-chain fatty acids, because the short-chain fatty acids are going to move straight into the blood rather than being esterified. It's like a longer process. So we know that the long-chain fatty acids are re-esterified, but what actually happens? Well, the long-chain fatty acids in the intestinal cell, in the enterocyte, they're bound to fatty acid binding proteins. And this does three things. First of all, it prevents toxicity. It prevents leakage out back into the lumen or even across the basal membrane. And it ensures transfer to the smooth endoplasmic reticulum of the enterocytes. And what happens on the smooth endoplasmic reticulum is that the long-chain fatty acids are, as I said, they're re-esterified to triglycerides using 2-monoacyl glyceride or glycerol 3-phosphate. And these are like the diglycerides and that the fatty acids are re-esterified onto. And once they're re-esterified to triglycerides, they're then bound to something called apoproteins, packaged with esterified cholesterol and phospholipids. So the fatty acids, they're re-esterified to triglycerides, bound to apoproteins, and packaged with esterified cholesterol and phospholipids. So what does this create? Well, this creates a chylomicron. And what a chylomicron is, it's a lipoprotein, so it's lipids bound with proteins. Triglyceride bound with an apoprotein forms a chylomicron. And remember, this is still in the enterocyte, so the chylomicron is in the intestinal cell. And it goes to the Golgi bodies in the enterocyte where it's processed, and then it's exocytosed across the basolateral membrane to the lymph. From the lymph it enters the blood, and from the blood it reaches organs. So we can compare long-chain and short-chain fatty acids by saying, well, the short-chain, they move straight into the blood, whereas the long-chain fatty acids, they're re-esterified to triglycerides, bound to apoproteins to form chylomicrons, and they leave the enterocyte as a chylomicron, which is a lipoprotein, rather than just as a short-chain fatty acid. And we can summarize that here. So the chylomicrons are processed, they move to the lymph, blood, and then to organs. And when they reach the organs, well, what happens to the chylomicrons? Well, the exported chylomicrons from the enterocytes, they reach the endothelial surface of organs and tissues. And there's an enzyme, there's a membrane protein enzyme called, on the surface of the endothelial surface, called lipoprotein lipase. Remember, chylomicrons are lipoproteins. So lipoprotein lipase breaks down the triglycerides in the chylomicrons. And what you're left with is fatty acids and glycerol. And the fatty acids and glycerol, they're stored in the muscle and adipose tissue. And you're left with remnant chylomicrons. And remnant chylomicrons, well, remember, chylomicrons are lipoproteins. They're triglycerides, apoproteins, as well as esterified cholesterols and phospholipids. So you're left with, I guess, esterified cholesterol, phospholipids, some lipids, and apoproteins. So these are the remnant chylomicrons. And where they go is they go to the liver. So we can summarize that here. The chylomicrons are digested by lipoprotein lipase on the endothelial surface of tissues and organs, and they're digested into fatty acids, glycerol, and remnant chylomicrons, which go to the liver. And these remnant chylomicrons that go to the liver, they're endocytosed into the hepatocytes, the liver cells, and they're converted to VLDLs, which are very low-density lipoproteins. And the purpose of VLDLs is to transport cholesterol. So the remnant chylomicrons are converted to VLDLs by hepatocytes for the transport of cholesterol, which is produced by the liver, remember.